Good morning, good afternoon, and good evening, everyone, depending what part of this globe you are from. I would like to welcome all of you to the 2022 CITAC Best Paper Award in Metrology award lectures. So without further ado, I would like to invite the chair of CITAC, Dr. Bernd Güttler, to address us all and present a welcome lecture. Thank you. Bernd, the floor is yours.
So CITAC, the mission of CITAC is, CITAC is a corporation that has been founded in 1993 in conjunction with the Pittsburgh Conference on Analytical Chemistry, and aims at a cooperation on international traceability in analytical chemistry. So the aim is and the mission to improve traceability on the results of chemical measurements everywhere in the world, and to ensure that analytical measurements made in different countries and at different times are comparable. And that was absolutely new at that time. And it was, of course, resembling that what has been done in the physical area in conjunction with the Meter Convention in the past as well. So this was a new step. And at that time, in 1993, the Consultative Committee for Chemistry and Biology was just appearing on the horizon and was starting its work. And those who have been involved in CITAC at that time, so people like Bob Karls and others became of eminent importance in the Consultative Committee for Chemistry and Biology later on as well. So he, Willy May and others, and Wolfert Wegscheider were already in place at that time and starting their activities. So what are our goals? So if you look at this more carefully, you can see that we are going beyond that, what has been devoted as a goal for the Consultative Committee for Chemistry and Biology at the Meter Convention, namely to facilitate the practical realization of metrological traceability in chemistry in practice. That is, to develop concepts that can be applied broadly at analytical laboratories and to disseminate those concepts. So we are looking outside of the metrology world, of the National Metrology Institutes as well, and try to make sure that the results that are carried out in practice are also traceable to the SI. So that includes to foster collaboration on metrology and chemistry as a means of effective technology transfer and cost sharing, or to promote the metrological principles in chemistry through guidelines and other tools for analytical laboratories. And this is really a core piece of our work. And I'm showing you a little bit more about that in a minute. So we want to promote and harmonize quality practices in analytical laboratories and to stimulate education and training on metrology in chemistry. So we do that all around the world. This is, to say it again, one of the things that were new at the beginning of CITAC, and that has not happened before in the same manner. And our members come not just from Europe, but also from Asia and from America to a large extent, but also from Oceania and Africa as well. And these parts are increasing. So, and when you look at our members, then you can see that, of course, a lot of them come from National Metrology Institutes, but there are all the other partners as well that we desperately need to install metrology and chemistry on a global level into every laboratory, namely the academia for education, the research bodies, the industries, and international organizations and others who are also part of CITAC. So we, as I said before, have a lot of ideas about guides and published guides. And we do that as a normal thing in collaboration with others. And the International Union for Pure and Applied Chemistry is a central part of it. And Zoltán is also a very much involved person in the IUPAC as well. So one of the last things that we published were the IUPAC CITAC guide for the evaluation of risk and conformity assessment of a multi-component material or object due to measurement uncertainty. And that has been published in the previous year. The other partner that we closely collaborate with is Eurachem. So this is a European organization that has, to a large extent, similar goals that CITAC has on a global scale. And we publish a lot of things together. which you can all see on our web pages or that of EURAChem. And the last thing that we did was actually the EURAChem CITAC guide on the assessment of performance and uncertainty in qualitative chemical analysis. And this guide in particular has raised a lot of attention. And it was actually our colleagues from Singapore who suggested to make a workshop on it and teach the things that are given there in this guide in an online workshop. And this is one of the benefits that we have due to the Corona crisis, that we know much better how to perform a video conference nowadays, and that's what we did. And that was carried out then at the end of January, at the beginning of this year, and the aim was to assist laboratories in using the EURAChem CITAC guide on qualitative analysis in practice. And this whole workshop was divided in 4 sessions on terminology and content, quality chemical analysis, qualitative forensic analysis, and qualitative analysis in laboratory medicine. And all those who were the authors of this guide contributed to it and were actually giving contributions which were all recorded and given to the audience. And what you can really see is that by having a video conference, you can attract a large number of participants. You're not restricted by paying money for traveling or anything else. So we had over 500 participants from 75 territories and at least 300 attendees in each session. And that really demonstrates of how much interest this workshop was and how important it is to actually gather the information on traceability in chemistry and put it forward to the people who need it. So, and I'm happy to say that these recorded workshops are also still available. So when you go to the CITAC webpage, you can, under conferences and workshops, see the videos on this workshop, and you can still collect all the information that was given there. So this is a kind of work that we do, and we add to this the CITAC newsletter, which gives you information about CITAC and all stakeholders in metrology and chemistry in one place. This is a speciality here, that we put all these things from different sources together. And this includes the CITAC Best Paper Award winners, of course, and their honored papers. And this is what it is all about today. And I now pass back to Zoltán, and he can give us some more information about that. So this is just the last picture about our members meeting a year ago, which was also, of course, carried out online in a video conference, but you can see that it is working fine. So thank you very much about this, from this point, about CITAC, and now everything about the CITAC Award. Zoltán.
Thank you very much, Bernd. I would like to share my slides. Here you should have my screen, I believe. So I would like to then proceed and announce the winners of the CITAC Best Paper Award for the calendar year 2021 on papers published in the calendar year or accepted for publication in the calendar year of 2021. The aim of the CITAC Best Paper Award is of the nomination is to highlight 3 important papers in the field of metrology and chemistry published in peer-reviewed journals that are worthwhile to draw attention to because of their scientific important scientific content. IUPAC or CITAC Best Paper Award was established in 2007, and since that over 40 papers were awarded, authored by us, as Bernd alluded to, leading voices, then emerging, now leading voices in our chemical measurement science community worldwide. A total of 236 authors have been named as winners of these or co-authors of these papers and winner of these awards from 28 countries or 5 or 6 continents, depending how you count continents. So it's a well-distributed global venture. So without further ado, 2021 winners were 3 papers. The first one is published in the journal Molecules by a German research team from PTB and BAM, supported by a visiting scientist from Nim, Thailand. And the paper is dealing with combining isotope dilution and standard addition for elemental analysis in complex samples. And the authors were Christine Braukmann, Aksa Praman, Olaf Rienitz, Alexander Schutz, Prane Pukpatanachai, and Jochen Vogel. So congratulations, and I would be inviting in a few minutes the authors of this paper to present their award lecture. So the next awarded paper was a team from organized around the nucleic acid working group of BIPM CCQM, and the paper dealing with a metrological framework to support accurate and reliable and reproducible nucleic acid measurement. The authors were Mojca Milavec, Megan Cleveland, Yung Kyung Bae, Robert Vilgos, Maxim Vonsky, and Jim Huggett. A truly international team from Slovenia, the United States, South Korea, the BIPM, Russia, and the United Kingdom. And the paper was published just a few short months ago at Analytical and Biological Chemistry. The next paper, it's a primary, science contribution, and it's been extremely topical, dealing with a labor-free detection of SARS-CoV-2 Spike S1 antigen triggered by electroactive gold nanoparticles on an antibody-coated fluorine-doped tin oxide electrode. And this team is from India, and the authors are Akanksha Roberts, Subhashish Mahari, Deepshika Shahdeo, and Sonu Gandhi. And the paper was published in Analytical Chemica Acta last year. So without further ado, I would like to invite the PTB BAM team to present their award lectures on combining isotope dilution and standard addition. So I stop sharing. And I would like to invite Olaf to share your screen, and
thank you, Zoltán.
the floor is yours.
Yeah, can you hear me?
Yes, we have your voice. Yes, thank you.
Okay, very good.
Yeah, it sounds good. It's clear. Yeah.
Yeah, for an unknown reason, I cannot share my screen. I don't know why. Maybe I also have to join Gavin in his office, so it will become crowded there.
That's the presentation room in PTB today. Ricardo, is sharing enabled?
No, I don't think so.
Or maybe just authorize Olaf to do the screen sharing. Maybe that's the issue.
Allowed. There is something here that is allowing multi-pin. I don't know what is that, to be honest. I can make you co-host.
Yeah, maybe that would be the easiest. Make Olaf co-host.
Is the scenario improving, Olaf? So now you are also co-host. So you have.
No, there's still a message that I'm not allowed to. That I'm not allowed to share.
There should be a problem with your computer, probably. I don't know any permissions from PTB.
Maybe I can send someone my presentation and then.
Okay.
Email, or...
Just send it to me, Olaf.
Zoltán, was that you?
No, it was Gavin.
Gavin, okay.
Might be quicker just coming upstairs rather than.
Yeah, that's even easier. Have you access to 3.1? Or I put it on the department drive. 3 or?
Try alla, yeah.
Yeah, you know.
So don't worry too much about these delays. It would be edited out from the recorded presentation. So the beauty of
It should be now or appear in 3 alle.
Yeah, I've got it.
It's called CITAC Best Paper Award, PTB, Rienitz, and so on.
So can you share it, Gavin?
Yeah, I'm just in the process of doing that now.
Fantastic. Okay, so you have the button to do it. Good.
Yeah, while Gavin is trying to share, I can already start, I think. In the very beginning, please let me thank you on behalf of all my co-authors for this great honor to be awarded with this Best Paper Award. And yeah, this is about, as Zoltán told you already, this is about combining isotope dilution and standard edition, and the aim was to yeah, now I see the screen, yeah, to do elemental analysis, yeah, for example in trace levels, or when these are very demanding elements, especially in complex samples. And I will show you in a minute the theory behind it, and also 3 examples from 3 groups. Okay, next slide, please. Yeah, this is the basic principle of standard edition. It's always the same. You have a certain set of solutions that you have to prepare. And these are blends consisting of nearly the same amount of sample. And in this case, when we talk about isotope dilution spike, and in case of normal standard edition, the spike can be replaced by any internal standard, or you can completely leave it out. And then you add increasing different masses of a standard solution. This is the light blue color. And then the idea behind is you plot your data points on the x-axis. Yeah, you have a value that is in a certain way proportional to the amount of added standard. In former days, and in the textbooks, this is the concentration of the standard added, which is not valid any longer. It's just a quantity that is proportional to the added amount of standard. And the y-axis, this is proportional to the signal of your analyte that you're measuring. In our case, it will then be even a ratio. Okay. Please press another button, yeah. And then when you do it like this, and yeah, construct a linear line connecting these points and extrapolate this line, then the x-intercept will be or can be used to figure out the mass fraction of your analyte in the original sample, because the x-intercept is simply the y-intercept of your linear line divided by the slope of your linear line. And this, in turn, is proportional to the mass fraction of the analyte in your sample divided by the mass fraction of your analyte in your standard. You can rearrange this for the Wx that you're looking for, and then you are done in principle. Okay, next slide, please. Yeah, why would you do standard edition, because it's a lot of work. You have to prepare several blends. And the idea behind standard edition is that you calibrate within the matrix itself. So when you're looking at very complex matrices or also at low analyte contents, then the effect of the matrix will be the same in all these blends, and the effect will cancel out. This is mainly the reason why you're doing standard edition. Yeah, and now you also need a reason for isotope dilution, because this is even worse when it comes to the amount of work that you have to do. But there's a very strong reason for isotope dilution, and the strongest reason is that you're measuring signal ratios rather than absolute signals. And this will help you to get rid of effects of signal drift, noise, fluctuation, and so on. And also, the results of your analysis are not any longer affected by your sample preparation. You can lose material or whatever. You can store it, and the acid or water can evaporate. This won't affect your signal ratio or your isotope ratio, and in the end, your signal ratio, and therefore, the result will not be affected. This is the reason why you're doing isotope dilution. And when you now compare these two techniques, this should yield a very superior method. And this is what we did in the end. Okay, next slide, please. The standard edition has a certain history, and it haunts me for nearly 20 years now. And in 2006, we did the first publication. This was dealing with ion chromatography back in 2006. And this is when we said goodbye to the volumetric preparation in standard edition, which is still what you find when you look into the textbooks. So the very first step was to get rid of the volumetric preparation and replace it with a gravimetric preparation and formulate an exact equation with no assumptions or approximations. And then some years later, our colleagues from LNE rearranged this exact equation in a much smarter way. So this was the next improvement, and this smarter rearrangement of this linear equation opened up the way for us again in 2012 to account for the uncertainty of the standard, which was still not accounted for in the uncertainty evaluation especially. And then another problem was that we combined the gravimetric preparation also with an internal standard. And you will find in the textbooks, your internal standard must not be present in your sample. Yeah? This is a no-no. But with this new equation, we found that the internal standard can be in the sample. And this is even advantageous because the uncertainty will be decreased when you use an internal standard which is already present in the sample. So this was in 2012. Yeah, 1 year later, we were heavily involved in the Avogadro project, and one of the problems was to measure a blank of silicon traces in TMAH, which is one of the demanding elements I talked about in a complex matrix. And this is when Axel and I said, okay, let's combine isotope dilution and standard edition. And we played a bit around with this, and the years went by, and in 2016, so our colleagues from NIC published exactly this combination of isotope dilution and standard edition, and even better than we thought about it, because they had an explicit and exact solution for this problem. Yeah, we tried this with a very surprising outcome, and this is why we then worked further on this problem, and in 2021 came up with another approach for combining isotope dilution and standard edition, but we don't use an explicit simulation. And this, as I'm going to show you in a minute, has some advantages. Okay, next slide, please. Yeah, this is again the basic principle. You have the sample, put in a certain amount of sample in all your flasks, add some spike solution Y, and then add some standard solution Z and top it up with your solvent. So in this case, for example, you prepare 4 blends. Next slide, please. And this is already everything you need to do the approach. You will see there's the tare mass of your vessel, of your flask or bottle. Then you have the mass of your sample, which is now individual, rather than what you find in the textbooks. And then is the mass of your spike solution, MYI, and the mass of your standard solution, MZI. And then you top it up with the solvent. And you might think, oh, now he forgot the mass of the vessel and also the mass of the solvent, but I didn't forget these because all these masses cancel from the equation. And this is one of the charming aspects of combining isotope dilution and standard addition, because in this way you lose several of the quantities that usually influence your measurements. And then so you have 3 masses, and then in the end, what you have to do is measure these ratios of the 2 isotopes, number 1 and number 2. And from the colors, you can see one of the isotopes is mainly from the sample and from the standard, and only a small portion comes from the spike, and the other is mainly from the spike, and the small portion comes from the sample and the reference. And now you can see already the approach. You simply model the isotope ratio in the ith blend, and this is the amount of substance in the ith blend of isotope number 2 divided by the amount of isotope number 1. And you can further go into details. This is the sum of what comes from the sample and the spike and the reference and so on and so on. And now, next slide, please. And now about 8 to 10 pages are missing, depending on your font size. And then you will end up with the equation on the top. And you can rearrange it in a way that on the left-hand side, you will find something which is, as I told you in the very beginning, which is proportional to the signal or the signal ratio of your analyte. And on the right-hand side, you will find a constant. In this case, it's the slope of the linear line times the x variable, which is proportional. You can see this. It's mz in the numerator, which is proportional to the mass of added standard. And then there's again plus a constant, and in this case, it's the y-intercept of your linear line. And when you look at this and remember one of the first slides, you can rearrange the x-intercept from the linear line so that you yield the mass fraction of the analyte in your sample. And this is now the y-intercept of the linear line divided by its slope times the mass fraction of the analyte in your reference times a certain constant which accounts for differences of the isotopic pattern in the sample and in the reference. When you think of the usual case, when the isotopic pattern of sample and standard is the same, then this equation even simplifies, and you will lose not only when you look at the equation, not only the x variable gets simpler because the expression, one of the expression cancels from the equation, but also the final equation gets much simpler because you lose the correction factor for the different isotopic pattern in sample and reference. And this is the equation that we then used for our 3 examples. Okay, next slide, please. Yeah, this is just to summarize this. You have a linear line and you have a certain y variable with the ratio in the blends and then the X variable. And this is then the third line, shows you the model equation that we also use then for the uncertainty calculation. And in the small table, you will find the meaning of all the symbols. Okay, next slide, please. Yeah, when we talk about traceability, one of the main homework is always also the uncertainty. This is the model equation. And in this case, you have to consider the uncertainty of the mass fraction of the analyte in the standard, the uncertainty of the y-intercept and of the slope. And in our case, where the linear regression yields the slope and the intercept, and both are calculated from the same set of x and y values, also the intercept and the slope are highly correlated, and therefore we have to consider in this equation also the covariance of the slope and the intercept. Next slide, please. Now we have to decide, please press another button. We have to decide how to do this linear line, and we opted for the most simple way. We simply minimize the so-called residues, the deviations of the data points from the linear line, and we introduce absolutely no weighting. So every point counts in the same way. Okay, next slide, please. And when you do it like this, you have to minimize the residual standard deviation, which is the capital S. You derive it for the slope and the intercept, and these are 0 and so on. And there you see the 3 points. Again, 10 pages are missing, and you will end up with the y-intercept and the slope. Next slide, please. And again, some pages are missing, and then you can calculate also the uncertainty, or in this case, it's the uncertainty or variance of the y-intercept and of the slope, and then you can also calculate the covariance of both. Okay, and then you put it into into the propagation equation for the variances, and again some pages are missing, and you will end up with this equation for the relative uncertainty of the analyte mass fraction in the original sample. Okay, next slide, please. Yeah, these are now the examples. One from my group, the inorganic analysis, silicon traces in TMAH. Another from Johan's group, inorganic trace analysis, sulfur in biodiesel fuel. And from Claudio's group, speciation analysis, which was transferrin in human serum. And all these have in common that these are complicated analytes in demanding matrices. Yeah, and we applied multi-collector ICP in case of the sulfur and the silicon, and a triple quad ICP combined with an HPLC separation technique. Okay, next slide, please. This is example 1. This is what it looks like, silicon in TMAH, all points nearly on the line. Next slide, please. And this is another, the sulfur in biodiesel. And next slide, please. These are already results. So each point represents such a linear regression. And the big advantage of the sulfur in the biodiesel fuel was that this was also part of a CC-QM comparison, and the red lines represent the key comparison reference value. And as you can see, the results from our new technique are well within these results. Okay, next slide, please. Yeah, this is the transferrin in human serum. Please, again, the next slide, please. And in this case, Christine used a material which was at least to a certain degree certified, so that we were in the comfortable position to have some kind of reference range, and both samples are within this range. So also a very convincing outcome, I think. And the difference between these 2 points, I think, can be attributed more to inhomogeneity of the sample rather than the measurement method itself. Okay, next slide, please. Just some words about the explicit solution, because I'm very sad that, because I like explicit solutions, because they're usually much better than our approximation. But this is what such an explicit solution looked like. The problem is it's suitable for daily use. Next slide, please. In case of silicon, you can discuss about this. Yeah, the red points with the uncertainties were calculated from the explicit solution, and the black ones are from the linear regression. You can discuss, is the uncertainty more reasonable or not? Okay, but from the second example, it becomes clear. Please, next slide. This is just the result from the transferrin in human serum. When you use the linear regression, in the green border, 2.2 or 2.1 approximately milligram per gram. And now, depending, because Christina used 4 blends, and the explicit solution is already completely done when you have 3 blends. So you can select a certain combination from these 4 blends, just select 3 of them. And for example, when we selected blend number 1, 2, and 3, the result that we calculated was -10 milligram per gram. And then we selected the blends 2 to 4, for example, and we came up with -5 milligram per gram. And this is, as sad as it is, from theory, much better explicit solution yields in practice with these uncertainty, with the uncertainties of our input quantities delivered simply numbers that were not reasonable in this range. Okay, next slide, please. This is already the summary and also the conclusion. Yeah, we came up with a new approach to combine isotope dilution and standard addition, which is not based on explicit solution, but on a linear regression model. And we tested it successfully in case of 3 complex matrices and challenging analytes. And the explicit or analytical approach, so superior in theory, showed that there are limitations when we put in real-world data. But this is very sad, and we're still trying to figure out why this is the case. And I hope we will find someday a solution. Yeah, the regression model provided also reasonable uncertainties, as I showed you. And this is also already a look into the future. We extended our approach to organic IDMS in cooperation with some pharmaceutical manufacturer. And their lab already is using this example, and the new publication is already under preparation, and it will have also some more advantages that I'm not at liberty to disclose at the moment. Okay, but this is still an ongoing story. Okay, thank you very much for your attention. Gavin, please press again. Then, yeah. Okay, thank you very much.
Thank you very much, Olaf. The conference is open for questions and comments. Very interesting presentation. Thank you. So I would suggest that if you have a question, just unmute and speak up.
So Olaf, I've got one, if that's okay.
Yep.
Normally, many of us who started using IDMS and exact matching IDMS in the first place started to do this because the uncertainties associated with linear regression were normally too high.
Yeah.
Yeah. So at the moment you're using a 4-point linear regression, where the degrees of freedom is normally n-2 on the number of points. Yeah, so you've got 2 degrees of freedom in this. So how does the uncertainty really come across as so low in comparison to the exact matching methods?
Yeah, the number of it's not only the number of points, but it's the number of point times the repetitions that you do. So it's the number of points that define your linear line.
So
if you measure
each of those points has multiple measurements.
Yeah.
Okay.
In case if you repeat it on 5 different days, you will end up at, for example, 20 points -2. So the degrees of freedom is 18.
Yeah. Thank you.
So can you muse about Olaf? Just following up on Gavin's question, on overall uncertainty profile of this approach compared to exact matching ID, because you contrasted with the analytical solution, and it's clearly there was some issues there. But if you would contrast this, is this a potentially a superior approach or just a more efficient approach? Compared to the exact matching
IDMS, I don't think you can compare this, because you would use this standard edition IDMS approach when you have a very complex matrix. In the exact matching, you need to get rid of your matrix in the sample spike mixture, for example. So you have to do analyte matrix separations for the sample spike blend. And this is something which you simply don't need to do when you combine isotope dilution and standard edition. So I think this is a totally different approach with totally different use cases. When you cannot, or you're not willing to separate the analyte from the matrix, then this is the method of choice. Every time this is simple or easily done, then the exact matching would be the better method.
Thank you very much. Very interesting. Francesca.
Also, yes, hello, good afternoon. Congratulations for the presentation. I like it very much. I have a question from a statistical point of view, again on regression. You have been used ordinary least squares, so that means that the statistical model underlying is assuming that the y values have all the same variability, the same uncertainty. So a unique weight, as you said. But if you have some information on the uncertainty on the y value that could be reasonable, evaluated, and could be assumed to be actually different from in the range from point to point, you may be interested in applying, for example, in try to applying weighted least squares that also has a very easy and closed analytical form, so it's not complicated. And in a more general way, if you want to consider also uncertainty on the x value, if they are not negligible with respect to those in the y value, you might be using weighted total least squares. And in that case, you don't have a closed linear form, but there are already over there software performing the estimation and the uncertainty calculation. So I think it could be worthy of investigating how the final certainty would be different from the ordinary least squares. That's just a comment on future possible developments.
Yeah, you're completely right. But this was just some first approach to do the evaluation, because our focus was on the experiment.
Yes,
rather than on the mathematics.
Of course, but I'm a mathematician, so I was focused on that. So just a comments and a suggestion for future developments. But thank you.
Yeah, thank you.
Thank you, Francesca. And again, please join me congratulating the TTB BAM NIM Thailand team for winning this award. Thank you very much, Oris. You can use the emojis, emotion, signs below to celebrate. So there's no clapping in other ways. So thank you very much, Olaf.
Okay, thank you very much. You're welcome.
So our next presentation will be delivered by Dr. Megan Cleveland from NIST on behalf of the nucleic acid working group based team on metrological framework for nucleic acid measurement. Megan, the floor is yours.
Okay, thank you so much on behalf of myself and my co-authors for selecting our paper and give me the opportunity to present it. So let's see if I can share the screen. Let's see. Okay, we will do this one. All right, and then let's see if the presentation. Okay, is that okay? Can you guys see the screen okay?
Yes, we have your screen, Megan. Thank you.
Okay, perfect. So our paper is titled Metrological Framework to Support Accurate, Reliable, and Reproducible Nucleic Acid Measurements. So it's kind of an interesting paper because it doesn't present, a scientific method. It's kind of an overview of the NOG and nucleic acid analysis. So first I'm going to give a brief introduction to the NOG, and then I'll talk about what nucleic acid analysis encompasses. I'll give examples of nucleic acid measurements. I'll talk about progress with nucleic acid measurements, challenges with nucleic acid measurements, and then some examples of NOG studies. So a very brief history of the nucleic acid working group. In 1999, the CGPM realized the growing importance of biotechnology, and they established an ad hoc biometrology group. 2 years later, in 2001, the Biometrology Task Group became the Bioanalysis Working Group, which is called the BOG for short. Due to expansion of biotechnology and also group participation, in 2014, the BOG was split into 3 different working groups. So the NOG for nucleic acids, the POG for proteins, and the COG for cells. And so these 3 working groups are under the CCQM with the other CCQM working groups. So we have quite a lot of participants from at least like 25 NMIs and DIs, the last time I counted, that have participated in the last 2 to 3 years or so. So we have members from all over the globe. So what does nucleic acid analysis encompass? So it's the detection, identification, and the quantification of nucleic acids. So detection would be, is the target present? So for example, does the patient have SARS-CoV-2? Identification would be which organisms are present. So you could have, a patient who's sick, but you want to know, what virus or bacteria is causing the disease. You could also have a patient with cancer, and you could want to find out what mutations are present in the tumor sample. Quantification, as it would imply, is how much of the nucleic acid is present. And this can be either absolute quantification, so you could get something in, nanograms, or it can be copies per mil, or it can be relative quantification. So you could have, 1% of this food product is genetically modified. So the types of measurements that would be included under nucleic acid analysis would be anything having to do with chromosomes, so the karyotype, the structure, any translocations, insertions, deletions, duplications, anything with the epigenome. So DNA methylation is important for cancer measurements. Anything, of course, with the genome itself. So anything having to do with DNA sequence, where a base changes or a certain base needs to be identified. The total amount of DNA, which could be either in mass or in gene copies. And then things like length. So STRs are short tandem repeats. Those are measurements of repeats that are important for paternity testing and forensic investigations. Measurements of DNA damage, and then anything to do with the transcriptome and the RNA material. So what areas use nucleic acid measurements? They're widely used. So in medicine, of course, they're very important in infectious diseases, in cancer, genetic disorders for diagnosing all these patients. They're also very important for food safety. So, one area is microbial contamination. If you have some type of salad or something that's contaminated with salmonella, you can use nucleic acid measurements, typically NGS, to track down that outbreak. You can also use nucleic acid analysis for detection of genetically modified foods. They're also important for environmental monitoring. So you can use nucleic acid analysis to monitor waterways for contamination with fecal bacteria. And I think most people have seen in the news, there's also measurement of SARS-CoV-2 in wastewater to measure increasing or decreasing community levels of COVID-19. They're used in biotechnology for the characterization and optimization of production strains, and then in pharmacy, so for gene therapy, and then for measuring residual nucleic acids in a lot of these biologic drugs that are now available. So one of the main things that the NOG does is these inter-laboratory studies. These studies are intended to provide evidence of international equivalence between measurement services. And so following successful participation in a study, an NMI or DI can submit their calibration and measurement capabilities, which CMC is for short, for review. And if approved, the CMCs are entered into the key comparison database. But in addition, NOG is using these studies to establish the traceability of nucleic acid measurements to the international system of units. So I'm just going to briefly talk about PCR because one of the main measurement techniques that we use in the nucleic acid working group is a form of PCR. And if I had given this talk 3 years ago, maybe there would be some people who weren't familiar with PCR, but I think over the last 2.5 years with the pandemic, most people have at least heard of PCR. So basically, PCR allows us to detect specific nucleic acid sequences. And basically the way it works is that you have these primers, which are short regions of DNA that flank the region of interest. And basically by a series of heating and cooling reactions, every time you heat the DNA, the two strands come apart, and then you let it cool back down and the primers anneal, and there's a DNA polymerase in there that extends the template and basically makes a copy. And you just do this for a series of cycles, and you get a doubling every cycle. So at the most basic type of PCR would just be a qualitative PCR that you would run on the gel at the end, and it could tell you whether the target is present or not. But then in the 90s, there was a new PCR technology that came out referred to as real-time PCR or qPCR. And basically the way this type of PCR worked was that it would fluoresce when the DNA was present. You could either have a dye that binds all double-stranded DNA, or you could have a target-specific probe. But anyway, you would get a fluorescent reaction, and the more DNA that there was to start with, the sooner the reaction reaches the threshold that's set. So something that comes up at 21 cycles here is more concentrated than something that comes up at 25 cycles. So this was a relative quantification because you could tell that this one is more concentrated than this, but without some kind of calibrant, you couldn't do the absolute quantification with real-time PCR. So then I'm going to talk about the evolution to digital PCR. So digital PCR basically uses the same reactions as the same materials in the reaction as qPCR, except that the reaction is fractionated into many different partitions. And these can be either, droplets in an oil emulsion, or they could be physical chamber partitions. But in any case, the DNA is fractionated into all these partitions. And by limiting dilution, some of the partitions will have a target molecule and some will not. And so you run the cycles, however many cycles you're going to do. It's typically about 40, but we've gone up higher than that. And the ones that have a DNA molecule will fluoresce at the end of the reaction, and the ones that didn't have a target DNA molecule will not fluoresce. And so if you know the partition volume, which is typically given in microliters, you can, by looking at the fraction of the targets that were positive, you can calculate the concentration in copies per microliter without using an external calibrant. So this method has been really great for NMIs and DIs to certify our reference materials, because we can we just have to know the droplet volume, and then we can calculate a very accurate concentration on the material. So some of the challenges that exist for nucleic acid analysis. So nucleic acids, they start out in a cell, either an animal cell, a plant cell, bacterial cell, and they need to undergo extraction, which is the isolation of the nucleic acid from the host cell. And the extraction methods can be highly variable. They can vary up to a log. And so another factor is that DNA, while it's generally quite stable at low concentrations, you can have the DNA stick to the plastic tube that it's in. So if you have a very low concentration of DNA that you're going to store for a while, you typically need to add some type of carrier molecule, such as yeast tRNA or some other type of background nucleic acid, so that the majority of what sticks to the tube is the background nucleic acid, and not your target. So sometimes the original matrix can contain nucleases that can carry over during the extraction process and degrade the sample. This is particularly a problem with RNA samples and RNases. And then if the nucleic acid is fragmented, assays with a smaller target size will yield greater concentrations than assays with a larger target size. When the starting material is RNA, which is the case for SARS-CoV-2, the material has to first undergo what's called a reverse transcription reaction. And so the reason it's called reverse transcription is that we have the central dogma that DNA to DNA is replication, and then you get transcription where DNA is transcribed into RNA, and then RNA is translated into protein. So these reverse transcriptases are able to take RNA and take it back to DNA to convert it to cDNA, which is what's required for the DNA polymerase to extend the material for the PCR to work. And different reverse transcriptases can vary in their efficiency. So that's another area that can result in bias or error. So let's see. So steps that may be required for nucleic acid analysis. So as I mentioned, extraction, reverse transcription. You could also have treatment with restriction enzymes, which is called digestion. If you're going to sequence the molecule, you often need to ligate adapter sequences onto there. And then if you're working with something like methylated DNA, you need to do bisulfite conversion. And all of these steps, add potential sources of error and or bias. So the one figure that we have in the paper talks about, part A talks about how the nucleic acid samples can vary from extremely simple to extremely complex. So at the most simple level, you could have a single type of DNA that is in some kind of aqueous buffer. So that should be, if the sequence is known, that should be a very simple nucleic acid to measure. But then you could have something extremely complex. So you could have something like, you could think of sausage, which is, it's a processed matrix with, it could be a mixture of genomes. It could be pork and beef and chicken, all that. and then it's in this matrix, right? So it's got fat and protein and all kinds of things that could potentially inhibit the reaction if they're carried over during the extraction. And then also the type of nucleic acid analysis can vary from extremely simple to extremely complex. So for example, the most basic one would be one target and just the presence or absence. So, SARS-CoV-2 would be like that, right? Like, is SARS-CoV-2 present? Just yes or no. And then you could have, the absolute quantification. So you could be trying to get the exact viral load for SARS-CoV-2 or any other virus. But then you could have something more complex where, you have a patient and that a patient who's sick and the cause is unknown. And so you might try and do some sequencing to determine what virus or bacteria or other pathogen could be causing the patient's illness. And then these are finally just some studies that we've done in the NOG. So the food area is more metrologically advanced. So a lot of the early studies were done with genetically modified foods. So in 2010, there was a genetically modified maize study. And then in 2015, we started doing studies that had different matrices with different properties. So in 2015, there was a GM rice study, which had a high starch matrix. And then in 2016, we did a GM rapeseed, which is a high fat matrix. And then currently in progress, there is a study which is measuring the percentage of pork present in beef, which is a high protein matrix, because all of these different matrices can have different, extraction methods, and they represent different challenges. So we're kind of trying to hit them all and do every type of matrix that you could possibly have. And then you would assume you would also be able to do, a moderate starch matrix, or moderate fat, moderate protein. And so some of the more recent studies have been health-related studies. So in 2017, there was the P184 study, which looked at mutations which are important in cancer diagnostics. And then in 2018, we did the P199 study, which was the copy number concentration of HIV. And then that was very timely because we had just done that study right before the pandemic, and it allowed us to quickly spin up P199B in 2020, which was the copy number concentration of SARS-CoV-2 RNA. So this is just some brief, the SARS-CoV-2 P199B study is still being written up for publication, but just a brief overview of the results. They were very good. So they were within plus or minus 40% of the mean, which may seem high for chemistry and physics, but for a lot of these diagnostic interlaboratory studies, they're really happy if things are within half a log, because some of the measurements, when they send out these samples for measurements, they vary by 2 logs or even 3 logs. So the fact that all these different institutes across the world could get within plus or minus 40% of the mean was considered very good. So finally, I just want to thank and obviously acknowledge my co-authors. So 2 of the co-authors, so Jim is the NOG chair and Maxim is the NOG vice chair. And then in addition to all the NOG members, especially Helen Parks, who was the former chair of the NOG. And this is the last picture that I have of the NOG together, taken in September 2019 at the meeting in Italy. So that's it, and I'd be happy to take any questions.
Thank you very much, Megan. Very interesting presentation, and congratulations to you and the team to the award. The lecture is open for discussion. Please unmute and ask your question. Megan, if you would take down the screen, that would be great, so we could see each other. Thank you. Thank you.
Yeah. Let me just figure out how to do that. Okay, let's see. I see pause. Okay, stop share. Got it.
Fantastic. Thank you very much. So maybe I start the questions. Can you talk about not maybe during these COVID times, or maybe in more general, how the nucleic acid working group activities and the standardization efforts and traceability efforts relates to the wider, world of clinical and food testing in terms of organization? So do you work with IFCC and the EUACs or codexes of the world to flow all these metrological knowledge into their practices? So is there some kind of formalized interaction?
Yeah, Jim might be able to answer that better than I can. But do you want to take that one, Jim? I mean, I would just say we have member overlap.
So where NAVAC fits into the big picture of nucleic acid measurements in the world, yeah.
I mean, NORG directly interacting with the IFCC is I mean, I'm on the Committee for Molecular Diagnostics. Well, I'm a consultant on the Committee for Molecular Diagnostics of the IFCC, and so there's a lot of communications there. As for formal interaction, I think other than via perhaps the JCTLM, where we're trying to implement reference measurement systems in nucleic acid analysis. Again, it's slow progress. Presence of NORG members on ISO, both in 276 and 212 molecular diagnostics and microbiology. We were pretty instrumental in the SARS aspects. There was a fast, and Megan was part of that fast track technical specification, which had, metrological traceability, the need for reference measurement systems. Again, NORG members are also on, I forget which working group it is of 212, but involved in, and Helen involved in the 17511 update, which included molecular considerations like the digital PCR. And I guess driving other areas like with the WHO, the international standards route for the virology. Megan and myself and others are there's getting much better cross-pollination in there now than perhaps there was a decade ago. As a formal, I don't, any kind of, other than that, I don't think, we're just fighting the good fight, Zoltán.
That's all what we can ask for. Thank you very much, and congratulations for the NAVAC team for winning this award. I can see Bernd coming on the screen. You have a question?
Yeah, first of all, thank you very much for this excellent presentation. And I think it is worthwhile mentioning that this has been a very fast result for a reference method on nucleic acid determination and quantification, actually, in a very, very difficult area, where we were struggling with for many years. And this is now a result which seems to be really helpful. And I mean, the obvious application to this is, of course, to use it as a kind of reference point for any kind of COVID testing. So we need to know very much about the qualities of the different antigen tests that we have. Do you think that this method can be an application to give us information about the quality of the antigen tests?
Certainly, you could measure the samples that go out in the antigen test also with PCR, and you could compare that to, you could get a absolute quantification on the copies per mil, but you'll still have the extraction method, which the antigen tests, there could be some difference in the extraction method, but certainly you could use it to benchmark things that go out in an antigen proficiency testing scheme.
Yeah, thank you very much.
Could I add as well to that I think at the moment the community is comparing RNA-based measurements to inform protein-based measurements, and whether that may or may not be appropriate. But at the moment, the RNA-based measurements they're using with the qPCR is calibrated rather chaotically, even if it is calibrated. And so there's this kind of fog of information as to whether that would be appropriate or not. And so the digital PCR would hopefully provide a much more accurate route to answer that question.
Yeah, that's what I think. Yeah, thank you for this effort.
Thank you again, and please join me congratulating the winning team. Congratulations. So our next presentation for today and from the third winning paper of 2021 is by Professor Sonu Gandhi, talking about labor-free detection of SARS-CoV-2 Spike antigen. So who would be presenting?
Hi, I'm Akanksha. I'll be presenting today.
Okay, Akanksha, the floor is yours.
Thank you. Is the screen visible?
Yes, we have your screen. Thank you. Thank you.
Okay. Hello, everyone. I am actually a fourth-year PhD student, and I work under the guidance of Dr. Sonu Gandhi at National Institute of Animal Biotechnology in India. And first of all, on behalf of all my co-authors, I'd like to say this is a real honor to have received this award for our work, and I'm really glad to be presenting it here today. And so without further ado, so our work is basically, we have made an electrochemical sensor, an FTO-based sensor, using SARS-CoV-2 spike antibodies for the detection of SARS-CoV-2 spike antigen. So, okay. We've all faced really hard times during this pandemic, and a lot of the research work that we've been carrying out, especially my PhD work as well, all of us at some point, especially working in government institutes, we all got invested into certain COVID projects. So the outbreak that happened in 2019, it basically brought the world to a standstill. And the causative pathogen, which was obviously identified as a new type of coronavirus and was renamed by the World Health Organization as the 2019 novel coronavirus. It's led to approximately 6 million deaths worldwide, more than 5 lakh deaths in India. And the issue with the diagnostics in this case was there were varied symptoms, a huge range of symptoms. So just based off of the symptoms, it was not, could not be predicted whether it was COVID or not. And so now the virus itself is a single-stranded positive sense RNA virus, around 30 kilobase pairs in length. And it consists of 4 structural proteins: the spike protein, envelope protein, matrix, and the nucleocapsid proteins. And the shape also, this beta coronavirus has a crown-like shape, mainly because of the spike protein on the surface. And the reason I'm speaking so much about the spike protein is it is our target protein that we have targeted in this work for the detection purposes. And the spike protein, it consists of 2 subunits, S1 and S2. And it's mainly, the virus uses it for attachment, fusion, and entry. And it is this particular protein that the virus keeps mutating as the new mutations keep coming out to escape the immune system as well as the vaccines we're creating. So the S1, it basically, the S1 of the spike protein binds to the receptor binding domain. And this is how the virus enters into the cells. The SARS-CoV-2, it affects the epithelial cells of the lungs and causes massive apoptosis. And the viral infection and replication in the body, it causes inflammation, acute lung injury, as well as secretion of a cytokine storm of interleukins and interferons. And a lot of research has been carried out into the vaccines. And as we know, the mRNA vaccine, which both Pfizer and Moderna have developed, it's basically where the genetic material in the mRNA form it's inserted into the body, and so that the cells, the host cells themselves produce this protein or a piece of the protein, and and cause an immune response. Then there is the viral vector vaccine, which Oxford and AstraZeneca have come up with. And in this case, they use another viral vector, which in this case was adenovirus. And the virus attaches itself to the cells. It injects the DNA that we want to inject into the host cell to make the coronavirus spike protein, which triggers an immune response. Then we have a protein subunit vaccine, which Vovavax has made. And in this case, a part of the virus, that is a part of the spike protein, it stimulates the immune response in the body. And finally, the DNA-based vaccines, which Zydus Cadila is working on, in which the plasmid DNA, and it is basically, it is put into the DNA molecule is put in the plasmid, it induces a response against the virus. Finally, after all the research that is going on in vaccines, we come to the diagnostic side of things. So the problem with the issue with the vaccines is there are multiple doses required, booster doses required. So one of the biggest issues with COVID management was the diagnostic front, where we weren't able to do any mass screening of all the positive cases. And the earliest diagnosis was chest computed tomography, where serial X-ray images were taken, combined, so that we could get a cross-section image of the lungs. This was basically done to detect the typical radiographic features that the pneumonia that is caused by COVID-19 was showing. The issue with CT was that if a patient was asymptomatic or an early stage infection, so in that case, a CT would not be able to diagnose it. Then came the gold standard, which is even now used, is the RT-PCR, and which detects the trace amounts of the transcript, that is the nucleic acid, the envelope, and the DRP genes. And finally, the rapid antigen test kits, which we perform even at home by ourselves, the LFA-based kits, which detect the viral antigen biomarker. The problem, the limitations with most of these diagnostics is, one is they're time consuming, they're laborious processes. Especially at the height of COVID, we weren't able to get results fast enough. Labs were full. They were not able to go ahead with the RT-PCR because of the sample loads. And there are papers saying there are a few false negative results because of the low titer value. And so rapid mass screening was not entirely possible. Whereas the LFA-based rapid antigen testing kits, they do give an initial reading, but their sensitivity is low in comparison to the gold standard. So, which is why we took on the biosensing approaches. And in this case, the first thing that we looked at is the type of sensor. We have different types: electrochemical, optical sensors, piezoelectric, as well as thermal sensors. In our case, we have used an electrochemical FTO glass sensor. This was used as the working electrode. In a 3-electrode system, there was a platinum counter electrode and a silver reference electrode. All of this was placed in a redox buffer in a cell. And the detection was done using a potentiostat and voltammetry. And the reason we selected electrochemical sensors is they're ideal for infectious diseases. One reason is that they have high sensitivity, specificity, and they remain unaffected by sample absorbance or turbidity, in case the sample has any turbidity. Then second component was the signal enhancer or the amplifier. People use different kinds of nanomaterial. We stuck to citrate heat reflux synthesized gold nanoparticles. And the advantage of gold nanoparticles is they have enhanced conductivity. They're very easy to synthesize, one-step synthesis process. They're most importantly biocompatible with our bioreceptor that will detect the target antigen. And they form a stable and simple bioconjugation with the bioreceptor. And finally, the bioreceptor, which could be antibodies, aptamers, peptides, nucleic acid. We went ahead with polyclonal antibodies, which we raised against the SARS-CoV-2 spike protein. The reason we went ahead with polyclonal antibodies is it's cost effective. The generation time is less. You can easily generate it in mice as well as New Zealand white rabbits, which we used. They recognize multiple epitopes. So this gives them an overall higher binding affinity. So the sensitivity of the sensor increases, as well as they're able to detect very small quantities of the target antigen. And because they detect the multiple epitopes, they also have a higher affinity towards mutating antigens. So even if the spike protein, say, keeps mutating in the further mutations, certain certain epitopes will still be detected by the polyclonal antibody, which is why we went ahead with it. So this is an overview. And as you can see, the electrochemical fluorine-doped tin oxide FTO glass sensor that we used, it is modified with the AUNPs and which were conjugated with the SARS-CoV-2 antibody and used for the detection of the spike antigen. The LOD of this particular FTO sensor was found to be 0.63 femtomolar, and it was in the concentration range of 1 femtomolar to 1 micromolar. The first step is the immunization. So in the immunization, what we did is we took the spike protein, we immunized the New Zealand white rabbits, collected blood on different boosters, and separating the serum, we purified these polyclonal antibodies from the serum and conducted ELISA as well as Western blot to confirm. As you can see in figure A, the two bands, the 25 kilodalton band and 50 kilodalton band in the SDS weight gel of the purified antibody. Furthermore, when we conducted a Western, the 75 kilodalton spike protein, that is where the antibody went, this confirmed the binding of the antibody we've made with the SARS-CoV-2 antigen, spike antigen. This was the different boosters that were given, and we took the experiment up till booster 3, where a dip was observed, and we terminated the experiment there. Furthermore, we conducted ELISA, a binding ELISA as well as a competitive ELISA to determine the binding affinity. And through ELISA, the binding affinity was found to be 7.5 nanogram per ml. Now we had the next step where we had to synthesize the AUNPs, which we used a combination of the Turkiewicz and Friends of heat reflux citrate reduction method. And the antibody was conjugated via physisorption, direct adsorption onto the AUNPs. So as you can see in the UV spectra, cold nanoparticles gave a characteristic peak at 520 nanometers. And this, upon conjugation, increased to 529 nanometers as the size increases of the entire conjugate. Furthermore, even extra additional peaks were observed upon conjugation in the FTIR, such as the SH peak. Then we carried out even the hydrodynamic diameter was checked out on using the DLS. And the AUNPs gave an approximate diameter of around 21 nanometers. And upon conjugation, this increased to around 30 plus or minus 5 nanometers. And finally, we tested the zeta potential, again using the DLS. And in the zeta potentials it was seen, the AUNPs, because they are citrate capped, they have a negative charge of minus 42 millivolts. And so bare AUNPs are highly stable in a colloidal form. Upon the conjugation of this positively charged SARS-CoV-2 antibodies on top of the AUNPs, there was a shift towards a positive at minus 39. However, since this is still in the negative range, the entire conjugate was stable within the colloidal solution, which is what we required. And now comes the design and the fabrication of the sensor. We first took the bare FTO electrode. So the glass electrode, one side is doped with FTO, and that is the side, the conductive side. On top of that, we drop cast AUNPs, deposited the AUNPs. And finally, we added the antibody, left it overnight to conjugate on top of the AUNP. Furthermore, this was washed, and then the electrode was used for detection purposes. For the detection, we took the spike antigen, and we taking a range of different concentrations of the antigen, we spiked it in buffer samples, and we used to detect it. And we got the different voltammetric graphs to see it. Okay, the optimization of the FTO electrode. The first is the layering experiments. As you can see, the red line, that is the bare FTO, giving a current around 2,500 microamperes. This drastically increased when we added the AUNPs, that is this dark blue line. So addition of this AUNPs increased the conductivity of the electrode. When we immobilize the antibody on it, there's a drop or a decrease that was observed. This is due to the protein masking effect that happens. So as the antibody covers the surface of the conductive surface, less electrons are transferred, and which is why there's a drop. And as further antigen attaches to the antibody, further drop can be, decrease in the current can be seen. So once the conjugation was done, we went ahead with detecting which was the best concentration of antibody to go ahead with in this particular experiment. As you can see, as we increased it from 0.25 to 1.5 microgram, the antibody gave the highest peak at 1 microgram. So we went ahead with 1 microgram of concentration of antibody for fabrication of the electrode. We also checked different scan rates, and the scan rate increased, the current increased with increasing scan rate. So we went ahead with 0.1 volt per second as the scan rate for all the experimentations ahead. Response time of the electrode was also checked, and it was found that 10 seconds and beyond, a stable reading was observed by the electrode, which is why this particular sensor could detect the antigen within 10 seconds. Finally, the environmental factors such as pH and the temperature were also determined. And the pH, the best pH that gave the best current was at 7.5, and a little decrease at 7 as well, which is perfect for any kind of physiological sample. And also we tested the different temperatures, and the electrode worked best at room temperature as well as at 37 degrees. So we went ahead with the experiments at room temperature. Once optimized, we did the analysis of the different concentrations, as I mentioned earlier. So different concentrations of the antigen were spiked in buffer, and they were detected on the electrode. And as you can see, starting from 1 femtomolar at the highest, as the antigen concentration increased, the current kept decreasing, like I mentioned earlier, with increased protein masking effect. The standard calibration curve was plotted, the log was calculated, and using the LOD formula, that is 3 into the standard deviation of the response divided by the slope, we got an LOD of 0.63 femtomolars within this range. Finally, it was also checked for any cross-reactivity, for example, with closely related species such as the MERS antigen, avian influenza virus antigen, and the HIV antigen. And these were spiked in saliva samples. Since detection of the spike protein would benefit if we were able to do it ultimately in saliva samples, as that's a non-invasive method. So we went ahead and spiked these around 1 micromolar of all the antigen in saliva samples, and we used the sensor to detect it. As you can see in the case of SARS-CoV-2 spike, there was a drop in the current, but no major drop was observed in either MERS, AIV, or in HIV. Similarly, this fabricated electrode was also used on multiple different samples to see if it could be repeated, and the same electrode could be repeated up to 4 times before it had to be washed and refabricated. And lastly, we also did a stability test to see how long the fabricated electrode could be used for testing if stored at 4 degrees Celsius. And it was found that around the 4th week, there was a drop in current found. So for approximately 4 weeks, this electrode after fabrication could be stored at 4 degrees. So finally, concluding, we use the SARS-CoV-2 spike antibody as a bioreceptor, and we've made an electrochemical immunosensor using gold nanoparticles as a conductive nanomaterial to detect the SARS-CoV-2 spike protein. And it has shown ultra-low detection at 0.63 femtomolar LOD in the linear response range of 1 micromolar to 1 femtomolar, and the rapid response within 10 seconds. Besides sensitivity, it was also highly specific for SARS-CoV-2 spike protein. When compared with the other viral proteins, it could be used, repeated up to 4 times, and stored for 4 weeks at 4 degrees Celsius. Future work that we are working with is our lab has also designed a similar FTO electrode. We used gold nanorods in that. It's also a recently published paper in Biosensors Bioelectronics. We've developed it to detect SARS-CoV-2, and we've successfully detected in clinical nasopharyngeal swab samples in VTM and compared it to RT-PCR data, since that is technically the gold standard. And the sensor, hopefully we'll be able to further miniaturize it and in the future use it for rapid on-field point-of-care mass screening. As right now, it needs the potentiostat in the laboratory. Hopefully we'll be able to make it into a point-of-care detection technique. Thank you, and I'd like to acknowledge the National Institute of Animal Biotechnology, where I'm working, my supervisor, Dr. Sonu Gandhi, for giving me the chance to work on this project, and my colleagues, my lab members, and of course the funding agencies, that is Department of Science and Technology, SERB India, Department of Biotechnology, and CSIR India. Thank you very much.
Thank you very much, Akanksha. Very nice presentation, and congratulations to you and your team for the award. The lecture is open for questions and discussion. Oh, we can see Professor Gandhi on the video.
Hi.
So, Jim.
Hi there. Yes, thank you very much for the presentation and some of the stuff we're working on. I call this disruptive future technology that will put the rest of us out of business. my question is linked to the actual the equivalence with other measurements, be they lateral flow tests or molecular, in terms of a viral burden amount. Forgive me if I missed it, but do you have an idea of how many, I guess it's RNA copies that is the standard that's used, but roughly how many RNA copies per mil this biosensor is able to detect in terms of limits of detection?
Ma'am, should I go ahead or would you like to?
Yeah, you can answer. Yes.
Okay. Like I said, the second paper that we are currently, we've worked on, we are developing another FTO sensor to be able to detect on real nasopharyngeal swab samples. So right now, when compared to the RT-PCR data, we haven't done a quantitative analysis, but qualitatively, all positive samples, 100% have been detected. Whereas the negative samples, there is around 20% or there's 40% of false positive results, but we haven't done a quantitative analysis as of now with the copy numbers.
Thank you.
But we're working on it.
If you needed any any help at all with the RNA quantity side of things, we'd certainly be happy to assist in that, because as I alluded to when in the previous lecture, the use of qPCR has been somewhat chaotic, and lost 10 years of virology, but yeah, really good work, and I wish you the best of luck. Thank you very much.
Thank you.
Thank you, Jim. I have also a quick question for the antigen calibration. How you source the standards? So are there reference materials in that space, or so where where you calibrate, because you were claiming detection capabilities on moles and yeah.
Is this when we spiked the antigen, the different spiking of the, okay, fine. So there has already been papers done on FTO based sensors which use different ranges of different antigen detection, and for FTO sensors, even our lab, we're working on other viral antigens as well, and this is the based on the other reference papers, this is the range that is selected for the detection for the on the FTO in the molar range.
Okay, so the standards are probably commercial standards.
Yes.
From SARS
yes.
and other.
Yes, the spike antigen that we've purchased it commercially.
It's commercial sources.
Yes.
I understand. Okay, thank you very much. I can see Bernd coming back on camera. Please.
Yes, so thank you very much also for this very nice presentation, which adds another angle on the recent developments and the recent needs that are related to the global pandemic crisis. And I think this is at the heart of the matter of most things, and that's what we are all thinking of, and it is something that also hits us every day, every single one of us, if we have to do any tests related to the disease. So every progress is very much welcome, and it is very good to see that there are activities all around the world to help to solve this crisis. And I think that's very important, and we all have to cooperate on a global level to get this all sorted out and have an improvement on that. And I think this is what all our interconnections are about as well. So it is my very pleasure to thank you all for your excellent contributions. And I think it, in spite of some little technical hassles that we had, shows that this kind of concept for having a meeting and celebrating our award winners is a very good one, and helps us to get connected all over the world, not just in a single meeting in a single room. And so I think we will proceed with this kind of frame for the presentation of these activities, no matter whether we will still have the pandemic ongoing next year or not. So thank you very much to all of you all around the globe for all your presentations, for all the contributions that you have made to this seminar, and most of all also to Ricardo da Silva for organizing this all, because without him we would have not been able to do this. And so I think this is also good for us at CITAC to be visible all around the world. So thank you very much to all of you for all of this, and hope to see you again next year at a similar meeting with ever new research results on metrology and chemistry, and hopefully with a pandemic that's decreasing and gives us more freedom to interact and travel around the world as well. So thank you very much to all of you. Bye-bye.